So the first brave soul is Ravi Jandhyala, who's the Chief Medical Officer from Evidence for Access. And he's going to talk to us a little bit about registries and big data in rare diseases. So take it away.
Okay, first of all, I've got a request from my wife. She says, can she have that bell when you finish with it, so I can
negotiate
something. So, what I'm going to do really is I'm going to try and do as little sort of statement making as possible and try and lead the discussion for you guys to take away, because I can't possibly cover all this in 5 minutes. Essentially, what I want to do is to try and contextualize this in the discussions we've had already. We have a receptor, we have a list of products, medicines that will work on a particular receptor. What I want to try and understand and explore is, you tell me about the patient to whom that receptor belongs, because it's so easy for us to just say, right, you've got a label now, you've got a diagnosis, that's it. It's not the end of the story, because we know that every patient is different. And that's made really, really clear, and we suffer this every day. If there are clinical trialists out there, they will suffer this every day. RCTs, why do we need them? The regulators mandate them. And why are they a really big pain in the you-know-what? Well, because, especially in rare diseases, we don't have many patients to start off with. We are struggling with subject numbers. They're usually too something. They're too tall, too short, too heavy, too light. There's something the matter with them that means that we struggle to meet inclusion criteria. There's a huge amount of experience and potential subjects for study that just get discarded, and we don't really have that luxury in rare diseases. High attrition rates once they get screened, they start on treatment, something might happen to them which means that they fall out and they become lost as well. Long duration. Now, the smaller the population is, the longer it takes to recruit, means the actual length of time that the study takes to complete is. So, the final judgments, what we do is we have a trade-off, we have a smaller number of patients, and we actually have to make a decision on how good a patient is in the patient population, sorry, how good a drug is in the patient population based on a very small amount of experience. So, what we're doing is we're literally looking at a fraction of the patients out there. So there's real-world evidence, real-world experience. Essentially, physicians can do pretty much what they like, frankly. And if they're responsible for a rare disease population and they feel the drug will work, then I think they can pretty much fill out as many bits of paper as they want and actually get that treatment for their patient. That patient probably will not make it into an RCT. That experience, we may be running the risk of losing it. So by having a lot of really detailed information about patients in RCTs, it's great, but we're potentially losing information that is in the real world experience setting. So why do we need to worry about RCTs in rare diseases specifically? Well, I take my experience from pulmonary arterial hypertension. When I joined industry for being a plastic surgeon, I was looking after Libris, and we launched that in PAH at GSK. Looked at the studies, 120 patients. I was launching a different product in post-MI patients, that's 20,000. So literally, I'm struggling. How is it we can get a similar level of experience or understanding about these products? But it's all about how the study is designed. Essentially, what really jumped out to me. What's jumped out to me and what I can't forget is the two graphs. I haven't got them here, but placebo and then on treatment. Okay, randomized controlled trial. Patients are randomized to either receiving treatment or placebo for as little as 12 weeks. Okay, the patients who received the placebo arm then transitioned onto active treatment are never caught up to those patients who were on active treatment continuously, and that's bad. Okay, so we then get into a situation where placebo-controlled trials, especially in RCTs, potentially could be unethical. So we need to understand as well, how are we going to detect signals? How are we going to understand that these receptors, these targets actually really work with these new medicines, unless we try and have some sort of sensitivity around them? How do we pick up these signals that we're getting? MHRA, EMEA have very, very good adverse event reporting systems, yellow cards. I don't know if that many people use them, but they do collect serially, sequentially, cumulatively adverse events, so we understand the safety profile of our products. We should really have a way of capturing how well these new repurposed drugs work in the real world. Okay, with RCTs you're going to do, we need to understand how they work in the real world setting. I think that was alluded to by the previous speaker. So we need a target population that we need to understand. there will be data that will be collected in one shape or form, either a primary care database, either a disease database, or simply someone's notes that is written in a clinic setting. Oh gosh. So let's look at these three very, very quickly. Patient characterization, identification. So it's the owner of that receptor. PAH, you get a label. There are several different types of PAH patients. They could be males, females. They could be different racial origin. Okay. So essentially, with patient identification, I want to try and say, let's focus on the patient, not the label. Okay, they have a diagnosis. Let's understand more about them. What is it that makes them a patient? You know, some rare diseases will take 2.2 years to get a diagnosis. They'll see 4 doctors. Fabry disease, the average time to diagnosis is 19 to 22 years. That's a long time these patients are living with their disease. We need to understand how they're interacting with their disease, and that's important as far as the patient identification. As soon as we get them diagnosed, we can get them on treatment. We can stop this effect of them not doing as well as if they were identified earlier. Registries, we need to have disease registries, not product registries. We need to make sure that as many treatment options are set up in serial as possible. Maybe even a patient organisation can be the overarching owner of that registry and make sure that we publish those regularly. And finally, we haven't really got time to talk about this, but it's the clinical study data request system. So a bunch of pharma companies led by GSK have signed up to this I suppose initiative, which means that their CSRs, their clinical data registries, clinical data database, clinical study databases are then open to interrogation by interested third parties. They decide the protocol, which is then reviewed, and then that relevant bit of data is then open for investigation. So there is data that is out there in the form of RCT information that could be looked at again. Sorry, I couldn't do a better job at 5 minutes. I apologise. I will have that bell though because it works.